Hello, everyone. So, for today's video, we're gonna do the lecture study guide for the fourth test. So, I don't know how many pages of notes I got, but it did not take me nearly as long to get through it this time as it did the last time. So, with that being said, let's do this. Pardon my Swedish fish. So number one, we're looking at the classification PowerPoint. So when I reference slides, it'll be from this PowerPoint. So the differences between DNA and RNA. So DNA, deoxyribonucleic acid. RNA, ribonucleic acid. So the sugar is different from one to the other. So the sugar in DNA is deoxyribose, and the RNA sugar for the sugar backbone that it makes is ribose. So one is deoxygenated. DNA is double-stranded. 2 strands that form that double helix, where those bases pair up and it turns that traditional ladder spiral look. Double-stranded. RNA, single-stranded. One. Uno. There are base pairs that the DNA and RNA strands are made of. The DNA base pairs are adenine, thymine, cytosine, and guanine. And adenine and thymine always pair up in DNA, and cytosine and guanine always pair up in DNA. So A and T, C and G. That makes the word C and G look alike. So they will never pair up with anything other than A with T, T with A, T with G, and G with C in DNA. So the function of DNA, adjusting my blanket. I'm cold in my house, y'all. The function of DNA is to store genetic information. And then, so when you get to RNA, it expresses the genetic information. So DNA stores it, RNA expresses it. So it takes that information, synthesizes proteins and amino acids and building blocks and everything else. So it goes up from there. Another big difference between DNA and RNA, other than the one strand and the ribose versus deoxyribose, is the base pairs. So you still have cytosine and guanine, and they are always going to pair together in RNA as well. But you have uracil instead of thymine. So A and U will pair up together. So for all you Auburn fans out there, this one's for you, AU. So in RNA, it will be A with U and U with A. Now, if you've got the DNA, and we'll talk about this in a few minutes, the genetic code chart where you've got the strand of DNA and you have to transcribe it from the DNA to the RNA. So if you have TAC, that's going to be AUG. So if there's a T in the DNA, it's going to be an A when you line it up in the RNA. But if there's an A in the DNA, it's going to be a U in the RNA. Everybody good with that? All right, so next we are talking about the central dogma of molecular biology, which means DNA yields RNA yields proteins. So DNA contains the instructions, right? So if DNA stores that genetic information, it is the instruction manual of molecular biology. So it stores that genetic information and how things need to work and how they need to be expressed, or how they are going to be expressed in a certain individual. Because sometimes there are mutations and that's not good every time. So just FYI. But So DNA contains the instructions for making the proteins. What RNA then does is it goes in and it copies that information. So it gets that strand of DNA and copies it with its own base pair, and it pairs to it with an mRNA, so the messenger RNA. And it uses that instruction that it gets, it uses those instructions to make the proteins, to build the proteins. Two processes involved in gene expression is transcription, which is making RNA, so the RNA synthesis, and that's when you go from DNA to RNA. And then you have translation, so transcription, so think about when you transcribe something. Think about what scribes do in the hospital. The doctor takes notes, the audible notes, and then the scribe takes it from the recorder and types it, right? So they're transcribing those notes. And so the RNA is going into the DNA sequence and transcribing the sequence that it needs so that it can then translate that into making proteins. So you've transcripted DNA into RNA, and then you're translating the RNA sequence into proteins. So it's transcription, and then it's translation, which is the second process that's involved in genetic expression. And so translation is the protein synthesis, so you're making the protein. Polypeptide, same thing. The genetic code chart on slide 10 is important. So if it helps you, make yourself a long chain of DNA base pairs and then walk yourself through it. So on the slide, it has a list, a long strand of DNA. So I went through and I did that one, and as I pretend like I'm doing it with my left hand, like I'm left-handed, not even a little bit. So you go through and you take that DNA sequence and you match up the bases for RNA, because you're going from DNA to RNA. So then they're going to be in groups of 3. So you get it in groups of 3 as you transcribe those from DNA to RNA. And so those three base pairs are going to code for which amino acid you need. So on slide 8, there's a chart that you read from the inside out. So it's got the 4 base pairs on the inside. And so you're going to find which one the base pair starts with. And then there's a second circle, and you're going to find what the second one is. Then you're going to find the third circle, and you're going to find what the third one is. And then you're going to see what amino acid that codes for. So I would try to make flashcards of those. I don't know if she wants you to memorize those or if she's going to give you that chart. It would be truly lovely if she gave you the chart, because that's a hard something to memorize. But I would ask that question. If you're going to have to transcribe, then translate, you're going to need to know which 3 base pairs in the RNA together is the code for which amino acid. AUG is the start codon. So that means that you're going to start making those proteins. And then there are 3 different possible stop codons. The one that's listed, the one that's in the slide is UGA, which for majority fans out there, that's an easy one to remember too. That's why I can remember that one, because I'm a dog fan. So with that being said, on to the next. So slides 12 and 13, features and structures of viruses. So viruses are tiny infectious particles that can only reproduce by infecting a host cell. They are not alive. They cannot reproduce by themselves, which is that's a building block of life, is that you can reproduce by yourself. So you can't do that. Viruses can't do that. They have to have a host. So they don't really have cells. So they're composed of a nucleic acid and a protein, and they have to multiply inside the host. So specialized structures that viruses transfer their viral nucleic acid to other cells. So they have structures that they use to infect a cell and then input their DNA sequence in the cell's DNA sequence, and then when the cell is thinking it's reproducing its own stuff, it's really reproducing the virus's genome sequence, which they either are going to have DNA or RNA. They're not going to have both. That's another reason why they're not considered living, because they don't have both DNA and RNA. The structure of a virus, there's the viron, which is the entire viral particle of a nucleic acid surrounded by a coat. So, like, it's got the nucleic acid in the middle, which is the whatever, like, genetic material that it carries, and then it's surrounded by, like, protection. The genome is the genetic material, the nucleic acid. And it, like I said, it'll have DNA or it'll have RNA, but it won't have both. The capsid, which is the coat, the shell, the protection, the protein coat, it gives it its specificity. So that capsid is what makes the influenza virus, and human papillomavirus, the human papillomavirus, and HIV. It's what makes it HIV. Any virus you can think of, the capsid is what gives it its specificity to itself. They are composed of a capsomere, which is a subunit, and the capsid protein is always encoded by a virus genome. This makes sure that the virus and not the host provides the instructions for synthesis. So that is what gets in there, in that host DNA, in that host cell. And when the cell is replicating and it thinks it's just making new normal cells, it's really replicating viral cells. All right. Lytic cycle. I'm just going to kind of give an overview, and then I'm going to tell you to go look at those slides, because there's more specific stuff on there that I think you may need to look through. The virulent state, or phage. So this is in a bacteria. This is the lytic cycle of a virus that infects a bacteria. It's called a phage. Phage. The virulent phage infects bacterium. It hijacks the bacteria to make a lot of phages, and then it kills the hosts by exploding it, which is lysis. So it goes poop. So it's not going like this. It's not eating it. The virus has gotten in to that cell. So if this hand is the virus and this hand is the cell, the virus has gotten in there and it has done what it needs to do, and then it kills its host cell by going boop, and it explodes. The lysogenic cycle is the temperate phage. So it infects and inserts its DNA into the bacterial chromosome, allowing the phage DNA to be copied and passed along the cell's DNA. That's how that one works. And I look on slides 23 and 24 for that one. Next, we're going to look at bacteriophage and animal virus versus animal viruses. It says slide 19, but I don't think that's right. I found the information I needed on slide 29. So I would look at slide 29. But again, you might see something on slide 19 that I don't. But if you don't see anything on slide 19, look on 29. That could be just a typo in there. So the different stages of the life cycle of a bacteriophage, or just the different stages, and then we'll go through the difference. So you've got attachment, when the virus attaches to the host cell. penetration, when it gets into the host cell. Uncoating, which is when it sheds its outer layer and becomes its true self. Once it walks into the room, she gains some confidence and she's like, I am going to make sure everybody knows I'm here. Then you have biosynthesis or eclipse. Then you have chronic infection and then release. So in bacteria, the attachment uses tail fibers. The way that it attaches, it uses tail fibers to attach to the cell wall's proteins. Then for penetration, the viral DNA gets injected into the host cell. In bacteriophage, it's not required. There's no uncoating required. She comes in confident. She doesn't have to build that confidence and then shed some layers. She's already shed the layers before she gets in there. The biosynthesis in bacteria happens in the cytoplasm. The chronic infection in the bacteriophage is lysogeny. So it gets exploded. And then the release, the host cell is last. So that host cell gets gone. So in animal virus attacks, the attachment sites are the plasma membrane proteins and glycoproteins. So the proteins that are on the plasma membrane of a cell in an animal cell are where the virus attaches. And then for penetration, the capsid enters by fusion through endocytosis. So once that happens, the uncoating happens. So there's an enzymatic removal of the capsid protein. So there's an enzyme that sheds that coat, and then we're on like Donkey Kong. Biosynthesis in animal cells happens in the nucleus for DNA viruses, because that's where the DNA is housed, or in the cytoplasm for RNA viruses. So if the virus that is infected, if the virus that has infected has DNA, its replication in animal cells is going to be in the nucleus, which makes sense because that's where your DNA is. If it's an RNA virus, it's going to replicate itself in an animal cell in the cytoplasm. So once it starts biosynthesizing, once it starts replicating, then you've got the infection. You've got the chronic infection. It can be latent. It can not show up for years and years. Think about HIV. You can get infected and not know for like 10 or 12 years before you start showing symptoms. So that's why that one is super important, that information is shared. It's a slow viral infection. So like cancers, long-term, like think about hepatitis, and that can cause liver diseases for years and years and years. And we'll get into all that too. And then the release, the enveloped viruses bud out. So if it's enveloped, it buds out. If it's not enveloped, it ruptures the plasma membrane. So it goes pop. Okay. So next we're going to look at retrovirus and RNA transcriptase. So retrovirus, they have a reverse transcriptase enzyme. So the retrovirus will insert a DNA copy of its RNA genome into the DNA of a host cell. Sorry, my kid's coughing. He's asleep, but he got to go to the doctor tomorrow. We think he has asthma, but that's neither here nor there. So the retrovirus will insert a DNA copy of its RNA genome into the DNA of the host cell that it invades, and it changes the genome of that host cell. So retroviruses have a plus SS RNA. It's a plus SS RNA virus. It's the reverse transcriptase. And the reverse transcriptase uses this to make a complementary SS DNA. So SS RNA with reverse transcriptase goes to SS DNA. So that's where reverse transcriptase comes in, because usually we're transcribing from DNA to RNA, right? So now when that reverse transcriptase enzyme comes in, you're going from RNA to DNA. So the ssDNA is then made into dsDNA that integrates into the host chromosome and becomes a permanent part of the host. HIV is a retrovirus. So that's why it's never cured, because it gets into that chromosome. It gets into the chromosome, and that just permanently alters the DNA sequence because that viral sequence has gotten in there. So if there was a way to, I don't know, reverse the reverse transcriptase, it may, I don't know if that's even a possibility. I don't know. But an example of a retrovirus is HIV. So there's a name for that inserted viral genome, and it's called a provirus. This allows the virus to stay in the host for forever, long time, long time, and cause a chronic infection. That is on slide 43. So if any of that didn't make sense, I pulled that information straight from slide 43. Next, we're looking at examples of viruses that can cause cancer. So on 46, we start with the human papillomavirus, and that's HPV, and that causes cervical cancer. There are vaccines for that. Gardasil. I had my Gardasil shot. Right when it came out, I was in high school. I think late middle school, early middle high school, sometime. Anyway, yeah, it's really cool that there is a vaccine to protect against cancer. It's really neat. But HPV, the human papillomaviruses, because there's over 150 of these viruses that are in that classification. They are mostly spread through sexual contact. Look that up on slide 46 if you feel like you need more information. But HPV causes cervical cancer. There's an example. Next, we're looking at the Epstein-Barr virus, so EBV. That's the herpes 4 virus. That's mono. If any of you have ever had mono. When I was in the 6th grade, I had my tonsils taken out, and as I was recovering, came down with mono because it was opportunistic because my immune system was already low. It's passed typically through saliva. You have a higher risk of getting nasopharyngeal, so nose, throat, sinuses through here, nasopharyngeal cancer, Burkitt lymphoma, Hodgkin's lymphoma, or stomach cancer. So those 4 are related to the Epstein-Barr virus. And that's on slide 47. 48 talks about hepatitis B and hepatitis C, and those are viral infections that infect the liver and make you susceptible to liver cancer. Hep A can cause hepatitis, but it's not going to do anything long term. It's not with you long term like B and C are. A is what you get when you get a cold sore. B and C are when you're jaundiced and your liver stops working. That's typically like sharing needles, bad blood from a blood transfusion. I know someone who I work with at my other job. Her dad is a Vietnam vet, and he was in an explosion and had to have a blood transfusion. And back then, they didn't check the blood as stringently as they do now, and he got hepatitis tainted blood. And so now he has terrible liver problems and is on the list for a transplant. Also, heavy drinking will set you up for that too, just FYI. But that's also not here nor there. The Hep B and C are usually sexually transmitted, but they can also be other ways with bodily fluids. FYI. All right, so slide 53, we're looking at features of viroids. So viroids are smaller than viruses. They are covalently closed circular RNA molecules. So they are RNA molecules that are in a circle that are closed at the ends by covalent bonds. They have a rigid rod-like secondary structure, and it's about 250 to 400 nucleotides. They have no protein coat. They have autonomous replication and occur as circular and linear. So primary circular, secondary is linear. It forms with a high degree of base pairing while parasitizing the host's transcriptional machinery process known to infect only plants. So this is really only in plants. But it kind of hijacks the transcriptional machinery inside the cell. So, okay, next we're going to look at 56 to 58 features of prion. Features of prions, the earliest prion disease, the first prion disease in a human. So in slide 56, prions, Prions. P are proteinaceous, I infectious. Prions, prions. Proteinaceous, so protein, proteinaceous, infectious particle. They are transmissible spongiform encephalopathies, TSEs, and they are caused by like abnormal protein growth and coming together. Sorry, I'm checking the Brave score real quick. They're playing on the West Coast. They cause neurodegenerative diseases. They're very rare. Only 1 to 2 people per million per year are infected. People and animals are infected, and the infections can be infectious. They can be inherited. They can be spontaneous. They are incurable, and they are ultimately deadly. They are ultimately fatal. The earliest is called Scrappy. I saw it as Scrappy Dappy Doo from Scooby Doo. So it causes itchiness. The first one is Kuru, Kuru, yeah, Kuru. It's an exotic disease to this cannibalistic tribe in Papua New Guinea, and it's localized mostly to the capital FORE, 4 people, and who they marry and their offspring. But the earliest prion disease is the Scrappy, and then the first known human one is the Kuru. I feel certain that the tribe's diet has something to do with that. When you eat people, bad things happen. So, okay. On to the next question. What are CJD, BSE, and CW? Okay, so CJD is Creutzfeldt-Jakob disease. I call my husband Jakob sometimes just to be funny, but it's not spelled the same. It is, it is a PRPSC abnormality, and it forms aggregately on the brain. So it's like that protein buildup. It goes into this certain part of your brain, and it's bad. You usually don't last 4 to 6 months once you get diagnosed. It's a neurodegenerative disease. It's bad. Look at slide 61 for that one. For more detail. I didn't go into too much detail on these 3 diseases, but there are details on slide 61 for Creutzfeldt-Jakob, 62 for BSE, which is bovine spongiform encephalopathy, aka mad cow disease. So, bovine cow, spongiform encephalopathy, mad cow, encephalopathy, brain. It is a variant of the Creutzfeldt-Jakob, but it is what shows up in the cows. CWE, I think, is another typo. I think it's supposed to be CWD, because what I found was chronic waste disease. It affects deer and elk and reindeer, cicada and moose, the meese of the world. It is fatal to animals and there is no treatment. But again, slide 63 for that one. So, okay. Done with the first slideshow. That only took 30 minutes. The next three aren't really quite that long. Especially the next one. All right. Now we're on to pathology. So I did actually define the terms this time because I felt like the terms, the vocab, the definition, there it is. I felt like that was a little long. So I kind of shortened it to make it a little bit more streamlined. Okay. Localized or local is limited to a small area. Makes logical sense, right? Your hometown is local, right? Rome's local restaurants, Harvest Moon, Aventine, Blossom Hill. Makes sense? Systemic is where it's spread throughout the system, spread throughout the body. Focal is typically your teeth, your tonsils, which I don't have any, and your sinuses. So think about a focal point that people look at. When you smile, they look at your teeth and they look at your nose, and if your mouth is open, they're looking at the back of your throat. Primary, initial cause of infection. It's the first one. Secondary is an opportunistic pathogen that gets in there when your immune system's down. Subclinical and apparent. There's no visible, no noticeable illnesses, so subclinical. You're not seeing it, right? It's there, but you're just not seeing it. Sepsis. Sepsis es no bueno. Sepsis is a toxic inflammatory response from a microbial spread. Septicemia, blood poisoning. Bacticemia, bacteria in the blood. Toxemia, toxins in the blood. Virusemia, viruses in the blood. So look at the word. The beginning of the word is going to tell you what's in the blood. And semia is going to tell you that it's in the blood. Okay? All right. So slide number. Oh, no, we're still not done. Acute. Acute is fast in and fast out. An acute infection is limited to one spot. It's fast in and it's fast out. Chronic is long term. It develops slowly and it's not fast in and it's not fast out. Subacute is somewhere between acute and chronic. It's like you're not always sick, but you're sometimes sick, but it's not always there, but it's sometimes there, but it's not fast in and it's not fast out. It's somewhere in the middle. Next we have latent, which is the causative agent of something that can stay dormant in your body and then pop up. So think about the shingles. The virus that causes the shingles, it lives in your ganglion nerve and it will just activate and pop up and decide that now you're going to have the shingles. We're going to talk about that in a few minutes too. Okay. Next we have the chain of infection. So that is on slide 5. You have your infectious agent that gets in and creates a reservoir. So what is a reservoir? A reservoir is a banking system, right? So like it's like a storage that gets in and it creates a reservoir. So it's got something it can pull from at all times. So it is a continual source of infection. After the reservoir, you have to have a portal of exit and then a mode of transportation. And a mode of transportation must occur in order for viruses to spread. Or in order for anything to spread, really. And then after the mode of transportation, there has to be a point of entry into a susceptible host. So you get infected, and there's a continuous infection. and there's got to be a way to get the infection out. And once the infection is out, it's got to ride on something. It's got to find a car. It's got to find a vehicle of some sort. And then it's got to find a way in to a host that's susceptible to it. So that's the chain of infection. So slide 5, we're still on slide 5. The first infection, the primary infection, must come from the reservoir, because there's not a secondary way in at that point. It's got to be that initial one has got to come from the reservoir, that continual source of infection. The second, the individual must transmit it to others, either directly or indirectly. So directly, I cough on you, I'm sick, and I give you that. But I'm sick, I cough in my hand, and I don't wash my hands, I don't Germ-X my hands, I don't sanitize. I touch a doorknob, and then you touch a doorknob, and then you touch your face, or you put your hands in your mouth, or whatever. That's an indirect passing of whatever it is that I had to start with. Now, some things don't live that long outside of the host. Some do. It just depends. Next, we're looking at slide 6 and slide 9. So, direct transmission, indirect transmission, and vehicle transmission. Just kind of segueing in. So, direct transmission is person to person. Vertical, which is mom to baby, either through the placenta or through breast milk. Or horizontal, which is just other direct kind, or animal to animal, so like rabies, anthrax, that kind of stuff. Next, you have indirect, which is when an inanimate object, or fomites is what it's called, when the inanimate object comes into play. And it has to become contaminated, and then the susceptible host has to become contaminated from the contaminated object. Next, you have the vehicle transport or transmission, and it's transmission through vehicles such as water, food, air. So infected water, the water is the vehicle. Infected food, the food is the vehicle. If something is airborne and you breathe in the air that's infected, then that's how that goes. Standard precaution and transmission-based precaution on slide 8. So that slide that's in the middle there. Standard precaution is just the basic minimum precaution taken to prevent the transmission of diseases. So you wash your hands, you coughing etiquette, you cough into your shoulder, you cover your mouth when you sneeze, those basic things that we just do. That's that standard precaution, and it is not specific to a particular infection. It's just what we do. Now then you get into transmission-based precaution, and so in addition to the standard, you have specific things that you do. So for whatever highly transmissible something you got going on, it is specific precautions to that. There are 3 categories to this transmission-based protection. You've got contact precaution, which is used when the patient is infected with a disease that is spread by contact, whether that be fecal oral, whether that be sexually transmitted, whether that be any other kind of direct contact that you have with someone. Those contact precautions are going to be specific to that disease. So I keep going back to HIV, but it's one that everybody knows about, and it's one that is a really good example. So it is a sexually transmitted disease. If your sexual partner says that they have HIV, then A, you need to talk to the doctor before you do anything. B, you're going to have a barrier of some sort to prevent direct contact, because that's how it's spread. So level 2, you've got droplet precaution. So that's when a patient is infected with a disease that will spread by respiratory drops, but in a short distance. So like a mask or certain agents that are in the air that can kill something that might be in the air. So that is the droplet precaution. And then you have the airborne precaution, which is basically the same thing, but it's a little bit more intense, because the droplets is just a short distance. Airborne is when it can travel long distances in the air, so it's spread by the droplet nuclei or the aerosol, and it goes longer distances. So the precautions on that are a little bit more intense. All right, slide 10, we're looking at endemic, sporadic, epidemic, and pandemic. Again, this should be a review because we just did this on the last test, I think. An endemic is a disease that is consistently present in a limited region and then the spread is predictable. Sporadic is think about it logically. It's sporadic, it's irregular, it's in a few places, it's scattered or isolated, and it's hard to predict just because it's so sporadic and so not specific. Next, you have an epidemic, which is an unexpected rise in an infection, but in a specific region. So it's not something that you're overly stressed about it getting out. And then you have a pandemic, which we all just experienced a few years ago, a rapid spread, a rapid increase in a disease across countries and continents and, global, and the growth is exponential. All right, so slide 11. I'm not spending a lot of time on that. We did that last time. I'm trying to stay awake. Next we have disease development. Okay, so slide 11. We have step 1 is incubation, which is the time between the infection and the symptoms starting. So again, I keep going back to HIV, but think about HIV where you can be infected and then not know for years, or you can be infected with the flu and then within 12 to 24 hours you're showing symptoms. So each virus is unique. So the incubation period, so it's the time that it takes for the infection to get into your system and start replicating itself instead of your own cells. So that's why sometimes it takes a minute. So you were exposed to somebody with a stomach bug 24 hours ago, but you're not sick yet, but something's starting to feel a little funny. It took a little bit for that virus or that bacteria or whatever to get into your system to get going, but that's called the incubation period. So it's that growing in your system and then you're going to get it out. Next you have the prodromal. Hold on, let me pull it up because I can't read my handwriting. Sorry guys. I have to pull it back up because I X'd them all out thinking I had it all done. Prodromal period. That's what it is. Prodromal period. It's short and it's very early, and it's those general symptoms, those I don't feel so good, you're lethargic, you're tired, which is lethargic. You might feel a little weak, you might feel a little dizzy, you might have a little cough, or your stomach might just be like, something not right. Like, I should be hungry, but I'm not, and that's not normal. At least for me, I like to eat. So next you have the period of illness. So that step 3 in the development of a disease is the period of illness. And that's when the symptoms are the most severe. Your immune system may or may not be able to overcome the illness. If it is, great. That means you've been exposed before and you've got that immune memory to just go get it. But you may not have been exposed, and this may be your first exposure, and the next time you're exposed, you won't be having it as bad. But this time it's going to be rough. R-U-F-F, Darren Ruff. All right, so next is the 4th stage, which is the period of decline. So the symptoms are starting to get a little better. They're starting to subside. You're still vulnerable to maybe a secondary infection because your immune system is weak right now, but you're starting to get better. And then step 5 is the period of convalescence. So that's when you're recovering, the period of convalescence. That's when you're getting better. All right, so that's it for that slideshow. Next, we're looking at immunity. Immunity. All right, slide 3. Be able to explain why the immune system is described as a double-edged sword. So, good versus evil. Not really evil, but, whatever. Some people who have autoimmune diseases might argue with me on that, though. So, the positives, the good, it is your body's defense system. It is so, so important. Your immune system is so important to your overall health and wellness and quality of life. You can take vaccines to boost that system and be really good. Um, but it is so important for you to have a good immune system. Um, some of the negatives, you can have hypersensitivity. So, like, my daughter has really sensitive, like, skin and airways. So, anytime the weather changes, my son too, anytime the weather changes or gets a little bit hinky, their immune response in their airways get hypersensitive and they cough to get stuff coughed out. But once they start coughing, they can't stop. Um, they have some swelling in their airway, and so they have to have an inhaled steroid to make it get a little bit better. You have autoimmune diseases, which is when, so your immune system is really good at, or a functional, good immune system is really good at identifying self, which is your body, and then non-self, which is when something comes in that is not supposed to be there, it goes and gets it, right? That's the point of your immune system, is to protect yourself from things that are not supposed to be there. But with autoimmune, what happens is that your immune system has gotten has had a brain fart, and it can't decide if self is actually self or if it's not self and it needs to go get it. So autoimmune diseases are when your immune system starts identifying things that are yourself as things that are not yourself, and it attacks things that are perfectly healthy. POTS is autoimmune. There's a lot of thyroid diseases that are autoimmune. Autoimmune's not fun. There can be immunodeficiencies where you don't make quite enough white blood cells to go do what it needs to do. Sepsis, that whole trigger to a microbial infection that may send too much or might not send enough. Cancers, like, it can cause you to have a barrier to, like, a graft or a transplant. Like, that's why you have to go on immunosuppressors when you have an organ transplant, because a transplanted organ is not self. So if your immune system is fully functional and healthy, and you get a new liver or you get a new lung or you get a new heart, your immune system is going to try to attack that because it's not recognizing self there. And so you have to let your body you have to let that new part become part of your body so that your immune system can recognize it as self, so it doesn't reject the new organ. All right, slide 4, table about the immune system. That chart is muy importante. You need to study that chart. It's the lines of defense in your immune system. So you need to know what's the first line of defense. It's your skin. What's the second line of defense? What's the third line of defense? What's going to come up when, and what cells are going to get sent out when? So is it going to be a B cell? Is it going to be a T cell? Is it going to be a macrophage? Is it going to be an eosinophil? Is it going to be a neutrophil? Is it going to be the natural killer cells? Know that chart because it's important. Next we have innate immunity versus adaptive immunity. So innate immunity, you're born with it. It's not specific. It's just your immune system that you're born with. It's a quick response, but you don't have a memory. So, like, it's just like that standard practice of washing our hands and wearing a mask if we're sick and all that fun stuff on those precautions. So that's kind of what innate immunity is. So it's your factory setting. It's not specific. It has no memory. So it's going to be the same response each time until your body identifies what it is you've been infected with. And if you've seen it before, then it's going to send out the specific cells that it needs. But if it's not ever seen it before, then you're just going to get everything. You're just going to throw everything at the wall and hope something sticks. It is your first and second line of defense. So then you have your adaptive immunity, which is what you've acquired, immunity you've acquired through your lifetime. So a vaccine or getting sick, or herd immunity, herd immunity is so important. that's a whole nother tangent I could get on, but I'm not going to because we don't have the time. Adaptive immunity is antigen specific. So it has a memory. So it's slower, which is why it's your third line of defense, but it is specific to the antigen that it is going after. So if you have seen this before, if your body has been infected with this before, then your immune system has that memory of that, and it's going to send out what got it last time. Let's see. Where are we? Slide 6, types of immunity. You've got innate immunity, which we just talked about. You've got adaptive immunity that we just talked about. You've got the humoral immunity, which is the B cells. You've got the cell-mediated, which I think are the T cells. You've got active immunity, which is a natural immunity or an immunity through a vaccine. And then you've got passive immunity. So that is given from mother to baby, either through the placenta or through breast milk. Or you've got antibodies in a blood product that you've gotten, and those are passive immunities. What is phagocytosis? Okay, so phagocytosis is when a cell engulfs and digests large particles. So it's an immune response. You have these really cool immune cells that are called phagocytes. And so what they do is they see these large particles in the system that are not supposed to be there, and so they go, and they digest it, and they get rid of it. And that's an immune response. It's really cool. The steps of phagocytosis are on slide 7. So go look those up. Next we have inflammation. Prish. P-R-I-S-H. So what is inflammation to start with? Inflammation is a local defense response. It is bringing in the immune cells to destroy the injurious agent. So whatever hurts you, that inflammation is your body bringing in those immune cells to the right spot to trying to get rid of what has caused the damage. It's limiting the effects of the injurious agent by confining it to one spot and keeping its products in one spot. So it's confining the injurious agent to one spot and not letting it get past that one spot. It repairs and replaces the tissue that is damaged by that agent. Inflammation is like super cool. It is a really cool immune response. Okay, so now let's go to PRISH, P-R-I-S-H. Pain, redness, immobility, swelling, and heat. So pain. What is the cause of the pain? Redness. Redness is a blood flow. It's a increase in the blood flow in that spot. And so why are we increasing the blood flow? To get those agents, to get those immunoresponse agents to the spot, right? We want that blood flow to increase to get where we need it to be. Immobility. You've got pain, you've got swelling, or that causative agent is just keeping you completely immobile. Swelling. You've got a fluid buildup, or you've got swelling like an edema. It could be pus, it could be an abscess, which is never good, but if they're swelling, that's an immune response. Then you've got heat, which is an increase in cellular metabolism, which increases the blood flow, which again is getting that immune response to the spot where you need it. Next we have definition and function of histamine, prostaglandins, cytokines, and interferons. So I didn't define these, but on slide 10, you're going to have the function and source of histamines, cytokines, and prostaglandins. And then on slide 12, it's going to give you a slide about interferons. I didn't see like a technical definition, so I wanted you to look at those for yourself to see what you needed to know, because I didn't want to call down too much information and you not have what you needed. All right, so next we're looking at complement. What is the complement system? Complement is an immune response that marks pathogens for destruction and makes a hole in the cell membrane of the pathogen. So the complement system, it's chilling, it's going to poke a hole in a pathogen's membrane and just mark it for others to come get. So like, hey, come destroy this. Come get it. Come get it. Somebody come kill the spider. See the chart on slide 13. It's got a lot more information than that, but the overall definition of what the complement system is that it marks, it's an immune response that marks a pathogen for destruction, and it pokes a hole in the membrane wall so that if cells need to get in there, it can. Okay. Let's see. Function of erythrocytes, thrombocytes, and leukocytes. Erythrocytes, red blood cells. What do red blood cells do? What do they do? Tell me, class. What do they do? What do they do? What do they do? What do they do? I know I can't hear you, but I know what you're telling me. You're telling me that they transport oxygen, because that's the right call. Red blood cells, they transport oxygen. It's their job. They're good at it. Thrombocytes, those are platelets. You know what platelets do? What do platelets do? They clot. They keep your blood from just pouring out everywhere. So if you're on something like, I don't know, a blood thinner, you're going to bleed in a lot more freely or without restraint than you normally would if your blood had no blood thinner in it. So what a blood thinner does is it kind of suppresses those clotting factors in those thrombocytes. Sorry, something was happening on the baseball game. Next we have leukocytes. Leukocytes are white blood cells. And those white blood cells, they have a nuclei, or they have nuclei. They have a nucleus, if it's just one that we're talking about. But they are your immune system. They are your body's defense system. That's why if you're sick and they take your CBC, which is a complete blood count, CBC, complete blood count. Y'all are going to need to know that. When they look at your CBC, if your white count is above a certain amount, then that tells them there's something wrong. You're sick. You're infected with something. So yeah, not fun. Types of leukocytes and their number. So there's a mnemonic. Never let monkeys eat bananas. Okay? They may beg and they may plead, but never let monkeys eat bananas. Give a mouse a cookie and they'll ask for a glass of water. Don't let that monkey eat a banana. So you've got neutrophils, leukocytes, monocytes, eosinophils, and basophils. Never let monkeys eat bananas. Neutrophils, leukocytes, monocytes, eosinophils, and basophils. Okay? So they're numbers. What I would do is look at slide 18, but also I'm going to tell you. So neutrophils are about 60 to 70% of your leukocytes. The leukocyte cells are about 20 to 30%. So there's your majority. Those two, never let, neutrophils and leukocytes. Those are your two big ones. Next, you have the monocytes, and those are about 1 to 6%. Eosinophils are 1 to 3%. And then basophils are less than 1. Less than 1% of all of these cells. So, again, see slide 18. And I would make flashcards for this and the next. So, okay. Slide 17 is the types of leukocytes and their numbers. Slide 18 is for the next question. Know the function of the different leukocytes, so the white blood cells. So I would go on there and make a chart of their function and what they target. Because each cell does something different and they target different things. So when it's already in a chart, it's hard for me to call that down anymore. And if you've got the chart in front of you, I want you to access that chart. So go to slide 18, look at that chart, make flashcards. Next, we're looking at what is the difference between leukocytes and lymphocytes. Okay, so leukocytes, ready? These are T cells. So they're produced in the bone marrow, but they mature in the thymus, which is where the T comes from. They come from the thymus. They mature in the thymus of T cells. Thymus, T cells, thymus, T cells. Leukocytes, thymus, T cells. They are a cell-mediated response. They're an adaptive immune response, and the T cells attack a cell that's already infected. Next, we're comparing it to lymphocytes. And lymphocytes are B cells. So not only do they get synthesized and made in the bone marrow, but they mature in the bone marrow as well. So lymphocytes are B cells that are made and mature in the bone marrow. They are a humoral response. They are also adaptive depending on the antibody action in the cells. They're an antibody-mediated response. Not a cell-mediated response, but an antibody-mediated response. So B cells, they secrete antibodies. And the B cells use those antibodies on the surface of the cell to recognize what antigen it is, and they attack it from the outside of the cell. So whereas a T cell goes in the cell and gets rid of the infection that way, what a B cell does is it sees this, and it's going to send out something that will attack it from the outside. So B cells are on the outside, T cells go to the inside. Okay. Next, we're looking at name the 6 cells involved in innate immunity. Again, that chart on 19, it's going to tell you all that stuff. So if you've already made those flashcards, you already know this. But the 6 cells involved in innate immunity: neutrophils, eosinophils, basophils, mast cells, monocyte/macrophage, and the natural killer cells. Those are your 6 innate immunity cells. The next says the 6 cells involved in adaptive immunity. This is where I got a little confused, so that's why I said just know the chart. Go make your flashcards and know the chart, because I only saw 4. So, again, I could be wrong, but I only saw 4. So just go make those flashcards with that chart. The chart is a wonderful thing. Next we have the cells involved in humoral and cell-mediated. Humoral is B, cell-mediated is T. Okay? Slide 19, your best friend. Next, we're going to look at 21, about antigens. Antigen is really just a smushed together word for antibody generation. So, antibody generation, antigen. It is any type of marker that is recognized by the immune system. So, again, this cell has this marker on it. So this marker is going to signal to my immune system to send out this to get it. So it is immunogenic or non-immunogenic. So yeah, you're good on that. Let's see. What is a hapten? Antigen stuff is on slide 21, and I know it says that, but I gave you the definition about an antigen. That should be good, but if not, if you feel like you need more information, slide 21. What is a hapten? A hapten is a low molecular weight chemical that must bind to a carrier molecule to be antigenic. And the carrier is usually an endogenous or exogenous protein to which that low molecular weight chemical is covalently bound. So, things that are in a low molecular weight, which is less than 1,000 Daltons. So anything that's less than 1,000 Daltons that must bind to a carrier molecule in order to be considered an antigen. Okay? So the carrier is usually a protein that's either endogenous or exogenous. and it's going to be covalently bonded to that low molecular weight molecule. An example is urushiol. It's the oil found in poison ivy and poison oak. So it's got to have that oil in order to infect the person, to cause a response. I don't know if I said that right. urushiol. Urushiol. You know, I'm from Chautauqua County. Sometimes my accent's real hard to understand. Okay, next we're looking at MHC1 and MHC2. Let's see. MHC, major histocompatibility complex, is a collection of genes coding for MHC molecules found on the surface of all nucleated cells of the body. In humans, something bad must have just happened in the baseball game. In humans, MHC genes are also known as human leukocyte antigen genes, or HLA genes. MHC1 is found on all nucleated cells. Now remember, red blood cells don't have a nucleus, right? We Gucci? Yeah, red blood cells, no nucleus. So you're not going to find the MHC1 on it. It presents as normal or a self-antigen, and abnormal or non-self to trigger the T cells involved in the cellular immunity. Depending on what it needs, it's gonna present itself as one way or the other. MHC is found on antigen-presenting cells, or APCs, macrophages, dendritic cells, and B cells, and they're going to present abnormal or non-self pathogen for the initial T cell activation. Okay? So APCs, or antigen-presenting cells, are present antigens on the surface of their cells using the MHC2. Three of the three professional APCs are dendritic cells, macrophages, and B cells. Okay? Everybody good with that? All right. Next, we are looking at the functions of the three types of T cells. So the cytotoxic, the helper, and the regulatory. So cytotoxic directly kills the cell by binding to the infected cell, makes holes in the cell membrane, and the enzyme enters the infected cell is destroyed. So the enzyme enters and it destroys the cell. All right, so cytotoxic. So if it's toxic, it's going to kill it, right? Think about it that way. Helper T cells. They're the most important. They help in the activity of immune cells by releasing cytokines to activate the cytotoxic T cells. So they also activate the natural killer cells and the macrophages, and they activate the B cells. So those helper T cells are those, like, find the helpers, find the helpers. Those helpers are going to be, they help in all immune cells. And they activate the cytokines to say, hey, this is what we need here, this is what we need here, this is what we need here, and this is what we need here. Everybody needs to find the helper, right? Find the helpers and help the helpers. So those T helper cells, yeah, they go and assess the situation, they do the recon, and then they call back to base camp and say, we need a sniper on the roof, we need a team of 4 on the ground, and we need some backup about a mile away. So next you have the regulatory T cells, which they modulate the immune system. They are the main tolerance of self-antigens, and they prevent autoimmune diseases. Sorry, my husband just sent me a picture on my messages of what happened in the baseball game, and it was ugly. Okay, sorry. All right, regulatory, they makes sense, right? It regulates the immune system. And so it's the main tolerance of self-antigens, and it prevents autoimmune diseases. It's what makes sure that the immune system identifies self and non-self and attacks what is not self. Okay? So next, we are going to look at antibodies. Okay, antibodies. are immunoglobulins, or Igs. They are a Y-shaped protein, and it binds antigens and tags antigens for destruction by immune cells. So antibodies go up and say, you're bad, you're bad, you're bad, and you're bad, and you need to go away. So I'm going to call my friends, I'm going to call my big sister and say, this person was not nice to me, get rid of them. For all you big sisters out there, that one's for you. Okay, so next we're looking at mechanisms of actions of antibodies. So the opanic, slide 38. 38. Okay, know that one. Just make flashcards. Again, it's hard for me to make it any more clear than what the chart already makes it. So make your flashcards be your chart. You should be good. Okay. So slide 29, we're looking at the functions of the different antibodies. So the immunoglobulins. So you've got IgA, immunoglobulin A. It is the most common and abundant in mucus secretions. It protects mucosal surfaces, and it traps pathogens in the mucus, and it can't be digested. Next, you're looking at, I didn't put where these are like found, like where you can get them, like IgAs in dairy. Like I didn't put that in here, so you might want to go look at the slide 29 if you think she's gonna ask you like, what can you take or drink or eat or whatever to get this in your body. Like, I don't know if she's going to ask you that, so I didn't put it on here, but it's on slide 29 for some of them at least. Okay, IgD is mostly B cell bound or antigen receptor. It works with IgM, M as in mama, in B cell development. Okay, so IgE is the least common, and it's made by plasma cells. It is parasitic, so anti-parasites, and it causes the allergic reaction. Its FC region binds to B cells and mast cells. If you have an allergic reaction to something, the IgE is going to kick in. It's probably why it's the least common, because some people are allergic to everything, and some people are allergic to nothing, and some people are allergic to some things. So, IgG, so immunoglobulin G, is the major Ig in serums, so 70 to 80%. It is the smallest antibody, so it moves easily across the cell membrane. It's a good opsonizer, opsonizer, opsonizer. It is a moderate complement fixer. IgG3 is the moderate complement fixer. And it can cross the placenta. So because it's so small, it can cross those membranes. It's going to cross the placenta. So if mom has something that has IgG, it's good, and she's pregnant, it's going to cross the placenta and go to baby. IgM, M. What did I say earlier? I said M earlier. Oh yeah, because it worked with D. D and M, mom and dad. M is the largest antibody. It is first produced and secreted by B cells. It is your first response antibody. It is a good opsonizer, and it is a strong complement fixer. Your mama's going to give you good compliments if you earn it. So it is a strong complement fixer. So those are the different immunoglobulins, the Igs. The types of vaccines, again, slide 42's got those. Make flashcards, okay? You're going to need to buy a crap ton of flashcards. That's how you're going to get through this test, really, because, I mean, those charts make it pretty straightforward on this stuff, and I can't make it any more clear than what the chart already makes it, and I don't want to make it any more confusing. So, sorry, I'm having to scoot stuff over so I can see all of these questions. Okay, next we're on to microbial diseases, and this is the last slideshow. Woohoo! So I numbered these 1 through 25, and they are all fill in the blank. So what I'm going to do is I'm going to just read the question and give you the answer, okay? So, number one, two bacterial genera, genera that are frequent causes of skin-related diseases are Streptococcus and Staphylococcus. Number two, Staphylococcus aureus is the most pathogenic. It's a permanent resident of human nasal passages and easily transported to the skin. Methicillin resistant, causes of toxic shock disease, impetigo, folliculitis. When you think of staph, this is what it is. Staph aureus, okay? Okay. Why does my handwriting stink? The morble virus causes the measles. Reddish macules first appearing on face and spreading to trunk and extremities. The ruby virus, R-U-B-I, ruby virus, causes rubella, which is the macular rash that resembles the measles, which is the German measles. The human herpes virus, so HHV6 and HHV7, are the viruses that cause roseola, present in saliva of most adults, high fever followed by macular rash that lasts for at least a day or two. Number 6, the variola virus is the virus that causes smallpox, which was eradicated in 1980. Get your vaccine. Number 7, the varicella virus is what causes the chickenpox. The herpes zoster virus is what causes the shingles, and that's what lives in your ganglion nerve and it's just like chilling. It's chilling. And then it's going to go, hey, I'm here, and it's not going to be fun. The tinea pedis, feet, podiatrist, pedal, pedis. Tinea, T-I-N-E-A, pedis, either way you want to say it. It's what causes ringworm in the feet, so athlete's foot. Clostridium tetani is what causes tetanus. Clostridium botulism is what causes botulism. Clostridium words are hard. Perfringens is what causes gangrene. The Lissa virus is the cause of rabies. Streptococcal pharyngitis is what causes strep throat. Streptococcal strep, pharynge, throat, pharyngitis, throat. Okay. Let's see. Bordetella pertussis is the cause of whooping cough. Pertussis, if you've heard it called pertussis instead of whooping cough, it's Bordetella pertussis. Let's see. Name 4 bacteria that cause GI diseases. So diarrhea. Staphylococcus aureus, the Shigella virus, Salmonella typhi virus, and the Escherichia coli, E. coli, bad bacteria. Name 3 viruses that cause hepatitis. So you've got Hep A, B, C, D, E. Okay? A, B, C, D, E. Okay? A is the Picornaviridae. B is the Heptanaviridae. C is the Flaviviridae. D is the Deltaviridae. And E is the Calciviridae. So A, Picornaviridae. B, Heptanaviridae. That's a D, not a T. C is the Flaviviridae. D is the Deltaviridae. And E is the Calciviridae. Okay, so there's 5. Choose 3. Okay. Helicobacter pylori, H. pylori, can cause peptic ulcer. Treponema pallidum Treponema pallidum. Treponema. Thank you, Jacob. Treponema pallidum. He keeps me straight with my pronunciations. I butcher words. I know what they are, but I butcher them. Treponema pallidum causes syphilis. The herpes simplex virus 2, HSV2, causes genital warts. Genital herpes. I'm sorry. I got one ahead. Genital herpes is the herpes simplex virus 2, HSV2. 21. The papillomaviruses cause genital warts. 22. There are 3 types of anthrax: the cutaneous, or the papules, gastrointestinal, bloody diarrhea, or the inhalation, so septic shock. So when you think of anthrax, you think of the inhalation type, because that's what people sent in the mail. All right. Yersinia pestis. Y pestis, sorry. That is the cause of the plague, and its method of transportation is fleas and inhalation. Borrelia burgdorferi, or Borrelia mayoni. Those are the causes of Lyme disease, and those are ixodes, ticks. An infected tick is going to give you the Lyme disease. And the plasmodium, that is what causes malaria, and it is mosquito bites. All right, guys. That's it. That's all I got. So, hopefully this helps. If you have any questions, please feel free to email. Please feel free to whatever you need to do. Get in touch with me. Y'all's test is Thursday in lecture, I believe. So, good luck. Y'all are going to be awesome. Y'all are going to do awesome. Don't forget about your unknown stuff. So, if you have questions about that, ask your lab professor. That's the best I can say, since I have a feeling it's not just my lab that's going to be watching this. So just ask your lab professor about if you have any questions for your unknown. All right, guys. Have fun studying, and I shall see y'all tomorrow. Bye.